Chapter 11. Chapter 11, just as an overview, discusses how B cells are generated in the bone marrow. Um, and remember, when they come out of the bone marrow, they're programmed to recognize specific antigen epitopes. Then once they get activated, what is the activation process? What steps or signals are involved? And then with subsequent infections, you'll get differentiation. So you may start out with a B cell that produces membrane-bound IgMs or IgDs on their surface. But then once they get activated, some of their progeny, some of that number of plasma cells will go out, and then you have a number of memory cells. And some of those memory cells will actually differentiate to produce different kinds of antibodies. Sorry about that. Well, let me just start with the B cells. They mature in the bone marrow. They're naive or virgin B cells. You'll see them nowadays mainly referred to as naive B cells. And then they come into contact with the antigen. They go through differentiation. Many of those offsprings, the progeny from these, will become plasma cells. Plasma cells are fairly short-lived, etc. But then some of them become memory cells. And you can have some changes that occur in future generations of those memory cells, in that you can get switching from one type of antibody to another, so that you can undergo different types of antibodies. And so you can switch from having, down near the FC region, you can switch from having mu to gamma to alpha to epsilon, all of those different antibody isotypes in that bottom region. And that allows you to produce multiple different weapons against the same antigen. Now, you can also get affinity maturation. Not only are you using the ability of that B cell to recognize a specific antigen, or to produce an antibody that recognizes it, but you get better and better and better at recognizing those specific antigens. Now, normal B cells don't live long unless they find a home inside the Singles bar, which is the lymph nodes throughout the body. The ones that are flowing through the bloodstream, and there's a lot of them, then the ones that are flowing through the body may only live a few days to a week or so. In mice, I think it's 3 days. Here's a fact in your book. In mice, the recirculating pool of B cells is about 2 times 10 to the 9th cells. That's a lot of cells. So, a lot of times you hear of someone being immune to a disease. Well, that may be true because they may have B cells that recognize a specific antigen. Okay, but not everyone in a population necessarily has that. So you can go through and use antibodies that one person produced to give some passive resistance to some of these unusual diseases. Let's see. The total number of antibodies that you can produce are unreal. Well over 1 million different types of antibodies have been documented in a single individual. That in itself is pretty impressive. But when you combine it with the fact that they can use these in different ways, if you were to make an IgA out of an antibody, then that IgA carries out a specific function by being transported across mucous membranes. So, that's kind of an overview of what we'll be talking about in this chapter. But there's a lot of good stuff. We'll be talking about maturation, what causes the B cells to become active, what kind of signal pathways allow them to proliferate, the humoral response, which you remember is the non-cellular component of the blood serum. We'll talk about some induction for these humoral responses, germinal centers, and then how this response is regulated so that you don't get too much activation of the immune system, which could be bad as well. So that's an overview of what we'll be talking about in Chapter 11.